Hi, and welcome back to my YouTube channel. By now you all know me, but if not, my name's Mario. I put out videos primarily on clinical research, and this video is part of my study series on topics that are covered in the ACRP CCRA exam, or the Association of Clinical Research Professionals Certified Clinical Research Associate exam. So far, I have put out videos on ICH E11, dealing with pediatric populations, and ICH E9, dealing with statistics. In today's video, I'm going to talk all about ICH E8, or the general considerations for clinical studies. This is good general knowledge for clinical trials, so even if you're not studying for the ACRP CRA exam, this is stuff you should know or become familiar with if you're interested in clinical research. So if that's of interest to you, stay tuned. Before I dig into this document and cover general topics you should know and potential questions for the exam, I need you to take a moment and smash that like button and subscribe to my channel if you haven't done so already. The channel is working its way toward 2,000 subscribers, so thank you very much for your support. All right, so without further ado, I am going to go into this document and cover the highlights for the CCRA exam primarily, as well as things you should just generally know. So the first thing of note is the objectives of the document. So clinical studies of medicinal products are conducted to provide information that can ultimately improve access to safe and effective products with meaningful impact on patients while protecting those participants in the studies. So two things to note: safe and effective products and protecting patient rights. So that's what you're going to hear about in the guidance document. All right, so it does define here, for the purposes of this document, a clinical study is meant to refer to a study of one or more medicinal products in humans conducted at any point in a product's life cycle, both prior and following market authorization. All right, so one thing to note about clinical studies is before initiating a clinical study, sufficient information should be available to ensure that the drug is acceptably safe for planned study in humans. So you should have some data to back your giving the drug or using the product in humans, and you should also have pretty good data to support that it's safe. So you want to be able to both administer or use it and ensure that it is theoretically safe based on data you have. So the next section is scientific approaches in clinical study, design, planning, conduct, analysis. It says the essence of clinical research is to ask important questions and answer them with appropriate studies. Quality by design in clinical research sets out to ensure that the quality of a study is driven proactively by designing quality into the study protocol and processes. So the first thing to note here is just that the essence of clinical research: ask questions, try to figure out studies that answer them, and make sure that the questions are narrowly asked so that you can be pretty confident in the answers you get. And then just note that quality is designed at the very get-go from the study. And this could be a testable question of when you incorporate quality elements. And the answer is going to be you incorporate them proactively from the beginning of the study. All right, so jumping into the next section just talks about patient input into drug development, which says consulting with patients and or patient organizations during drug development can help ensure that the patient's perspectives are captured. This would make sense. You want to talk to the patients and patient organizations about things that you're designing for that specific population and get their input. So the next page talks more about quality, and it says quality is the primary consideration in design, planning, conduct, analysis, and reporting of clinical studies, and is a necessary component of clinical development programs. And then this is important. It talks about good planning and implementation of a clinical study, also derived from intention to design elements of clinical studies, such as the need for clear, predefined study objectives that address the primary scientific questions, selection of appropriate participants that have the disease, condition, or molecular genetic profile that is being studied, use of approaches to minimize bias, such as randomization, blinding, or masking, and or control of confounding, endpoints that are well-defined, measurable, clinically meaningful, and relevant to patients. The whole next section talks about critical to quality factors. So a basic set of factors relevant to ensuring study quality should be identified for each study. Emphasis should be given to those factors that stand out as critical to study quality. So there's a big emphasis on quality. And then the whole next section talks about approaches to identifying the critical to quality factors. I think you should just have some idea of the titles in the section. I don't see much being testable, but identification of critical to quality factors will be enhanced by approaches that include the following elements: establishing a culture that supports open dialogue, focusing on activities essential to the study, engaging stakeholders in study design, reviewing critical to quality factors, and considering critical to quality factors in operational practice. So those are your critical to quality factors. The next section jumps into drug development planning. This is more important in terms of the exams, and I'm going to read out the sentence here verbatim. It says, drug development planning should adhere to the principles of scientific research and good study design and ensure the reliability and interpretability of results. Efficient drug development includes appropriately planned interactions with regulatory authorities throughout the development to ensure alignment with requirements for product quality and to support approval in the condition or disease, including possible post-approval studies to address the remaining questions. For the exam, what is potentially testable here is when do you include regulatory authorities? And the answer would be you plan regulatory interactions with regulatory authorities throughout the study. So that's the only thing I see testable here, but just that's important to know that it's ongoing dialogue with regulatory authorities throughout the clinical study. All right, so jumping into the next section is the quality of investigational medicinal product. And again, an emphasis on quality, and you want to make sure that the products are what they say they are or claim to be. So they want to make sure that they're manufactured properly and that they're safe. So here it says, ensuring adequate quality and characterization of physiochemical properties of investigational medicinal product is an important element in planning a drug development program. Formulation should be well characterized in the drug development plan, including information on bioavailability wherever feasible, and should be appropriate for the stage of drug development and the targeted population. Evaluation of the quality of drug may extend to devices required for the administration and or companion diagnostics to identify the targeted population. All right, then we jump into non-clinical studies. So what do you need to know about non-clinical studies and what assessments does this incorporate? So it says the non-clinical assessments usually include toxicology, carcinogenity, immunogenicity, pharmacology, pharmacokinetics, and other evaluations to support clinical studies. So those are some of your non-clinical assessments. Assessments of the preclinical characteristics include physiology and toxicology effects of the drug, serve to inform clinical study design and planned use in humans. So this is some of the data you can use to assess safety prior to going into humans. So the next section is on clinical studies, and it says, although clinical drug development is often described as consisting of 4 temporal phases, phase 1 through 4, it is important to appreciate that the phase concept is a description and not a requirement, and that the phases of drug development may overlap or be combined. This makes sense. So when we think of clinical studies in general, we think of phase 1, phase 2, phase 3, or phase 4, but we don't necessarily have to have a study that's only phase 1 or only phase 2. You can have phase 1, 2, phase 2, 3. Generally, phase 4 will be alone because it's post-approval, but the definitions are not necessarily drawn in the sand, line in the sand. This is a phase 1 study. Sometimes you'll have a phase 1, 2 study or a phase 2, 3 study. Just note that. And I'm sure this document will go on to break down the definitions for the different phases. So this is of importance, and the use of biomarkers and why are biomarkers used. And the guidance says the use of biomarkers has the potential to facilitate the availability of safer and more effective drugs, to guide dose selection, and to enhance a drug's benefit-risk profile, and can be considered throughout drug development. Clinical studies may evaluate the use of biomarkers to better target populations more likely to benefit and less likely to experience adverse reactions, or as an intermediate endpoint that could predict clinical response. So that's why many clinical studies include biomarkers use. So the next sections break down the clinical trials and the different phases. So for your clinical studies, you have the human pharmacology studies, And the protection of study participants should always be the first priority when designing early clinical studies, especially for the initial administration of investigational product to humans, usually referred to as phase 1. So just know that's your definition of phase 1 trial. So it talks here, in phase 1, it talks about estimation of initial safety and tolerability. So studies typically include both single and multiple dose administrations, talks about pharmacokinetics, so characterizations of a drug's absorption, distribution, metabolism, and excretion continues throughout the development program, but the preliminary characteristics is essential early goal, and then it talks about pharmacodynamics. So PD data can provide early estimates of activity and efficacy and may guide the dosage and dose regimen in later studies. Next, we move on to exploratory and confirmatory safety and efficacy studies. So exploratory would generally be phase 2, and then confirmatory would be phase 3. So it says here, after initial studies provide sufficient information on safety, clinical pharmacology, and dose, exploratory and confirmatory studies, usually referred to as phase 2 and 3 respectively, are conducted to further evaluate both safety and efficacy of the drug. All right, so those are your definitions thrown in. So we've talked a little bit about that. So let's cover a little more on confirmatory studies. So confirmatory studies are designed to confirm the preliminary evidence accumulated in earlier clinical studies that a drug is safe and effective for use for the intended indication and recipient population. All right, so you're confirmatory, you're confirming that it is effective for use in the population that it's targeted for. And then confirmatory studies may evaluate the efficacy and safety of more than one dose or the use of the drug in different stages of disease or in combination with one or more other drugs. The next section is on special population. So some groups in the general population may need additional investigation during drug development because they have a unique risk-benefit consideration, or because they can be anticipated to need modifications of the dose schedule of a drug. And such special populations include investigations in pregnant people, investigations in lactating people, investigations in children, and just a note, investigations in children is all covered in ICHE 11, which I have a video out on, and then investigations in geriatric populations. So finally, we're going to talk about post-approval studies for these types of clinical studies. And just note that after the approval of a drug, additional studies may be conducted to further understand the safety and efficacy of the drug in its approved indication, and this is usually referred to as phase 4. All right, so jumping into the design elements and data sources for clinical studies. So study objectives impact the choice of study design and data sources, which in turn impact the strength of a study to support regulatory decisions in clinical practice. Clear objectives will help to specify the study design, and conversely, the process of specifying the design may help to further clarify the objectives. And then this is important. So an important distinction between studies is whether the allocation of individuals to the study drug is controlled by the study procedures, or allocation to the drug is not controlled, but exposure to the drug is observed in the study. And this is the difference between an interventional study and an observational study. So if you control the study procedures, you're dealing with interventional, and if you're observing, you're dealing with an observational study. So in more detail here, it says interventional studies, and in particular randomized studies, play a central role in drug development because they can better control biases. The design of randomized studies range from simple parallel group designs to more complex variants. And then it talks about adaptive design, which are studies that allow prospectively planned modifications to the study. It talks about a master protocol. So master protocol studies allow for the investigation of multiple drugs or multiple conditions under a shared framework. And platform studies. And so platform studies allow for multiple drugs to be investigated in a continuous manner with different drugs entering the study at different times and leaving the study based on pre-specified decision rules. So just know the definitions here: adaptive design, master protocol, and platform studies, and I think that's all that could possibly be testable. All right, so dealing here, it talks about special populations. The population to be studied should be chosen to support the study objectives and is defined through inclusion and exclusion criteria for the study. So talks about basically what type of populations, and says studies conducted in the earlier phases tend to be more homogeneous in study population, and studies conducted in the later phases of drug development or post-approval are often more heterogeneous in study population definitions. This makes sense because in your early phases, you're putting in inclusion-exclusion criteria and limiting to a certain subset, so you would get a more homogeneous population. And then as you expand and more population takes it, you'll get to a more heterogeneous population. The number of participants sample size in a study should be large enough to provide a reliable answer to the questions addressed. All right, so the next section is treatment descriptions. The treatments including controls under study should be described explicitly and specifically. And then it talks about your choice of control groups, and it says the major purpose of a control group is to separate the effects of treatment from the effects of other factors, such as natural course of the disease, other medical care received, or observer or patient expectations. So that is your control group. So comparisons may be made with placebo, no treatment, standard of care, other treatments, or different doses of the drug under investigation. So that's more into your control groups. And just know there are two types of control groups. There's internal or external control groups to the study. So this next section, and I'm just going to go through it quickly here, defines this. So it says the source of a control data may be internal or external to the study. The intent of using an internal control group is to help ensure that only differences between treatment groups are due to the treatment they receive and not to differences in the selection of participants, the timing and measurement of study outcomes or other differences. And it says with use of an external control group, individuals are selected from an external source, and the individuals may have been treated at an earlier time or during the same time, but in another setting than participants in the study. So those are your things to know, internal or external control groups. It says the use of external control requires that the disease course is well known and predictable. So if you're doing things outside of the study setting, it would make sense to make sure you know the course of the disease and it's well known and predictable. So that would make sense. Next section talks about response variables, and it says a response variable is an attribute of interest that may be affected by the drug. Study endpoints are the response variables for the that are chosen to assess drug effects. The primary endpoint should be capable of providing clinically relevant and convincing evidence related to the primary objective of the study. The secondary endpoints are either supportive measurements related to primary objectives or measurements of effects related to secondary objective, and exploratory endpoints are used to further explain or to support the study findings or to explore new hypotheses for later research. So just know your primary objectives, your secondary objectives, and your exploratory endpoints. The next section is all about reducing bias. This was covered pretty much in the video on ICH E9 and stats, but I'll just highlight a couple of things here. So it says the study design should address potential sources of bias that undermine the reliability of results. In studies with internal control groups, randomization is used to ensure comparability of treatment groups, thereby minimizing the possibility of bias in treatment assignment. Randomization at the start of the study addresses differences between the groups at the time of randomization, but does not prevent bias because of differences arising during the study. So just note, randomization can prevent bias from being pre-existing, but it can't prevent bias that happens during the course of the study. So this section, and I've started this, is important. It says concealing the treatment assignments limits the occurrence of conscious or unconscious bias in the conduct and interpretation of a clinical study that may affect the course of treatment, monitoring, endpoint, assertment, and participant responses. In a single blind study, the investigator is aware of the treatment, but the participant is not. When the investigators who are involved in the treatment of the clinical evaluation of the participants are also unaware of the treatment assignments, the study is referred to as double-blind. In an open label study, the consequences of lack of blinding may be reduced through the use of pre-specified decision rules for aspects of study conduct, such as recruitment, treatment assignment, participant management, safety reporting, and response variable assessment. So the reason I started this is, again, the definitions that you should be familiar with, which basically include the single blind, double blind, and open label studies. All right, the next section is more of statistical analysis. This, again, is covered in my ICH E9 video, but a couple of highlights. The statistical analysis of the study encompasses important elements necessary to achieving the study objectives. The specification and documentation of the statistical analysis are important for ensuring the integrity of the study findings, and statistical analysis of primary and secondary endpoints that address key study objectives with respect to both efficacy and safety should be described in the protocol, including any interim analysis or design or plan design adaptation. So again, this is just note for the exam. When do you incorporate the statistical analysis? It's going to be incorporated before the study starts and specified within the protocol. So it says here, for double-blind studies, the statistical analysis plan may be finalized before treatment assignments are revealed. All right, jumping on to the next section, which is on study data. It says study data comprise all information generated, collected, or used in the context of the study, ranging from existing source documents to study-specific assessments. And it talks about both primary data and secondary data, which you should be familiar with. So here it says study data can be broadly classified into two types: data generated specifically for the present study, primary data collection; data obtained from sources external to the present study, secondary data. So if it's coming from the study, it's primary. If it's external to the study, it's secondary. And it says a study may use both types of data. For all data sources, procedures to ensure the protection of personal data of the individual being studied should be implemented. The study protocol, and if applicable, the informed consent should explicitly address the protection of personal data. Data quality attributes include consistency, uniformity of assessments over time, accuracy, correctness of collection, transmission, and processing, and completeness, lack of missing information. So just a little more on primary. It says primary data collection, capture, and the subsequent processing provide an opportunity to prospectively ensure the quality of data. And it says secondary data use, the relevance of the available data should be considered and clearly described in the study protocol. Next section is on the conduct, safety, monitoring, and reporting. So it says protocol adherence to the study protocol and other relevant documents is important, and many aspects of adherence should be considered among the study's critical to quality factors. So an underlying theme of clinical studies is you want to write them in such a way that they can be followed, and there's an ongoing importance to the quality aspect. So individuals involved in study conduct should receive training commensurate with their role in the study, and the training should occur prior to their being involved in the study. It talks about data management here. It says the manner and timeliness in which the study data are collected and managed are critical contributors to the overall study data quality. And it says here, inappropriate access to data during the conduct of the study may compromise study integrity. So this is a little more on the common sense, and we want to try to minimize outside influences and just make sure we are paying attention to collecting data in a timely manner and emphasizing quality. All right, safety monitoring. So the goal of safety monitoring are to protect study participants and to characterize the safety of the profile of the drug. The approach should reflect the type and objective of the study, the risks to the study participants, and what is known about the drug and study population. Talks about withdrawal criteria here, so clear criteria for stopping treatment or study procedures for a study participant while remaining in the study are necessary to ensure the protection of the participant, but should also minimize loss of critical data. So you should have clear stopping rules. And then data monitoring committees. It says an important component of safety monitoring in many clinical studies is the use of an independent data monitoring committee. This group monitors accumulated data while the study is being conducted to make recommendations on whether to continue, modify, or terminate a study. And I've emphasized this when you're thinking about data monitoring committees in another video. just remember they are not involved in initiating the trial. Their scope is whether to continue, modify, or terminate a study. So an underlying theme or potential question just can throw you off is, oh, is the data monitoring committee involved in the initiation of trial? And the answer is going to be no. All right, so study reporting, clinical studies and their results should be adequately reported using formats appropriate for the type of study, interventional or observational, and information being reported. So consideration should be given to providing a factual summary of the overall study results to study participants in an objective, balanced, and non-promotional manner, including relevant safety information and limitations of the study. The transparency of clinical research in drug development includes the registration of clinical studies before they start on publicly accessible and recognized databases and the public posting of clinical study results. So reason I read that slowly is that's important. So how do you ensure transparency of clinical research in drug development? You make sure that your study is registered. We use basically a publicly accessible database to make sure that people are aware and can jump onto a website and see all the studies that are being run. So this is built into the recommendations, into the recommended guidelines. All right, so the next thing to consider here is just the final section here. So it is goes back to the types of clinical studies, and this is emphasized. So it says drug development is ideally a logical, stepwise process in which information from early studies is used to support and plan later studies. And it has a table built in here of importance. So it says, it defines the human pharmacology, exploratory, confirmatory, and post-approval, and the various objectives of the study. So I'll go over all of this because this is really important for your understanding of clinical trials and phase 1, 2, 3, and 4. So human pharmacology, also called phase 1, and the objectives of that study are to assess tolerance and safety, define, describe clinical PK and PD, explore drug metabolism and drug interactions, evaluate activity, assess immunogenicity, assess renal hepatic tolerance, assess cardiac toxicity. So that is your human pharmacology of phase 1. Exploratory, or phase 2, is to explore use for intended indication, estimate dosing regimen for subsequent studies, explore dose response, exposure response relationship, provide basis for the confirmatory study design, and then we can move into confirmatory or phase 3 studies, which demonstrate, confirm efficacy, establish safety profile in larger, more representative patient populations, provide an adequate basis for assessing the benefit-risk relationship to support licensing, establish dose response, exposure response relationship, establish safety profile and confirm efficacy in specific populations such as pediatric elderly. And then finally, your post-approval in your phase 4, which the objectives of those studies are to extend understanding of benefit-risk relationship in general or special populations and or environments, identify less common adverse reactions, and refine dosing recommendations. And that is it. That is everything covered in ICHE 8, the general guidance for clinical studies. In the next videos in the study series, I'm going to cover 3 more items that are on the exam, which include the Declaration of Helsinki, ICH E2A, and ICH E6. So look out for those videos. If you made it this far and you haven't done so already, please take a moment and smash that like button, and I will see you in the next video.